Welcome to the Deep Dive. Today, we're taking a really close look at plant genome editing using CRISPR-Cas9. It's a technology that's really changing things, food, health, even basic biology. Our mission today is to kind of unpack the complexity. We've got sources explaining not just the mechanics, but where it fits in agriculture, the global picture. We need to get the history of gene editing, what made CRISPR such a game changer, that RNA-guided bit, and frankly, what this huge power means for all of us.
Yeah, and it's really important, I think, to realize that cutting DNA, gene editing itself, that's not new. People hear CRISPR and think it just appeared. But the groundwork goes way back.
Decades, right?
Decades. We had tools like meganucleases, research started back in the 70s. Then zinc finger nucleases, ZFNs, they became more practical around, the early 2000s?
Okay.
And then just before CRISPR, in 2010, TALENs popped up. Transcription activator-like effector nucleases. Quite a mouthful.
And CRISPR-Cas9, the one that got the Nobel, launched all these companies.
Yeah.
That was just 2 years after TALENs.
Right.
So if ZFNs and TALENs could already do precise DNA modification, what was the big problem?
Yeah.
What held them back? Was it that protein-based mechanism?
It absolutely was. The hurdle was basically engineering effort, cost, and time. All those earlier tools, the meganucleases, ZFNs, TALENs, they all find their target DNA sequence using proteins.
Okay.
So if you wanted to target a different gene, you had to go back to the drawing board, re-engineer, test a whole new complex protein structure just for that one new sequence. It was, expensive, took a lot of work, and frankly, it was slow.
Right.
CRISPR just completely flipped the script on targeting. That was the breakthrough. Moving from protein guidance to RNA guidance, the Cas9 enzyme itself, the cutter, it's pretty much universal. It's the guide RNA that you program.
Oh, okay.
And that made the whole process way simpler, faster, cheaper. Suddenly, almost any lab could actually do it.
So it's like instead of needing a custom wrench for every single nut, you got one adjustable wrench and you just tell it which nut to grab. A huge shift in how it works.
That's a great analogy. Exactly. Yeah. And that big idea, it came from looking at nature. I mean, the story really starts in bacteria, not some high-tech design lab.
Right.
These weird repeating DNA sequences, clustered, regularly interspaced, short palindromic repeats. CRISPR, the first time someone noticed them was actually in E. coli back in 1987.
Wow, that far back.
Yeah. Imagine finding these strained, identical repeats separated by unique bits of DNA, the spacers. They had no idea what it was back then, but they'd found the heart of how bacteria defend themselves.
Must have just looked like genomic clutter at the time.
Yeah.
But later we figured out it's basically the bacteria's immune system, an adaptive one.
That's right. It helps them fight off viruses, phages, and those unique spacer sequences. Think of them like the bacteria's, molecular vaccination card. Right. When a virus attacks, the bacterium snips out a little piece of the virus's DNA and slots it into its own CRISPR array as a new spacer. It's a memory. And it passes that memory down. So
the specificity comes from that stored memory. The spacer sequence has to match the invader's sequence for the defense to work.
Exactly. And that's where the Cas proteins come in, CRISPR-associated proteins. They are the enzymes, the nucleases, guided by RNA copies of those spacers. The RNA finds the target, the Cas protein does the cutting.
Now, for the editing tool we use, Cas9 is the big name, usually from S. pyrogens. Why that specific one?
Well, there are different types of CRISPR systems in nature. Cas9 really took off for editing because it's efficient and simple. It's a class 2, type 2 system. That means it's a single protein doing the whole job.
One enzyme does the searching and the cutting.
Yes. Compare that to, say, class 1 systems, which need a whole bunch of proteins working together just to find the target site. Cas9 is neat, compact, much easier to get it working reliably inside more complex cells like plant cells or, human cells.
And for editing, researchers streamlined it even more, didn't they? Taking 2 natural RNA bits, the cRNA and tracRNA, and merging them.
They did. That was a key step for making it a tool. They fused the cRNA, which has the 20-letter address for the target DNA, and the tracRNA, which acts as a scaffold for the Cas9 protein to bind to, into one molecule.
The single guide RNA, or gRNA.
The gRNA. That's the elegant part of the engineered tool. It's this efficient combined piece that carries both the address and the handle for the Cas9 enzyme, directing it to cut the DNA right at that specific spot.
Okay, so that's the core innovation people talk about. The gRNA guides Cas9, makes the cut a double-strand break, and the cell sees that break as damage that needs fixing, right? How it gets fixed determines the outcome.
Precisely. The cell's own repair machinery gives us 2 main options. The first one, the most common, is nonhomologous end joining, NHEJ.
NHEJ.
Think of NHA as the cell's quick and dirty patch job. It just sticks the two broken ends of the DNA back together. But it's often a bit messy, not perfectly accurate.
So it makes mistakes.
It usually introduces small insertions or deletions, people call them indels, right at the cut site. And those indels typically mess up the gene's reading frame, so the gene gets switched off or knocked out. That's what you use if you want to disrupt a gene, maybe one that causes disease susceptibility.
So NHEJ is basically for breaking things deliberately. If you want to disable a gene, that's your go-to pathway.
That's the main use, yes. The second pathway is more like precision surgery, homology-directed repair, or HDR.
HDR.
Now, HDR usually only kicks in if we supply a DNA template, a piece of DNA, with the sequence we want to insert or use for correction. This template needs homology arms, sequences that match the DNA on either side of the break.
So you give the cell a blueprint for the repair.
Exactly. The cell uses that template to repair the break precisely. This is how you do gene correction or insert a specific new sequence. If you want to fix a faulty gene or add a beneficial trait, you aim for HDR. It's the difference between, say, cutting the wires to an engine versus carefully replacing a specific part.
That level of control and how accessible it's become, it's kind of mind-boggling. Our sources really emphasize that labs everywhere can use this now without needing, years and years of specialized protein engineering skill. That accessibility is driving all these applications. Let's touch on some of those, starting with maybe the most ethically charged one, gene drive.
Gene drive. Yeah, that's where CRISPR potentially moves beyond changing one organism to changing a whole population, maybe even a species.
Yeah.
It uses the Cas9 system to create what's called an autocatalytic mutation. It drives itself.
Autocatalytic, meaning it speeds up its own spread.
Sort of.
Yeah.
Here's how the basic idea, sometimes called a mutagenic chain reaction or MCR, works. Normally, an offspring gets one copy of a gene from each parent, right? 50-50 chance for any modified gene.
Right, Mendelian genetics.
With a gene drive, you insert your desired gene modification, but also the genes for Cas9 and the guide RNA right next to it on the chromosome.
Oh.
So when that organism reproduces, in the cells that form sperm or eggs, the guide RNA directs the Cas9 to cut the other chromosome, the one that doesn't have the modification.
It cuts the normal copy.
It cuts the normal copy. And then the cell's repair machinery, often using that HDR pathway we talked about, is tricked into using the modified chromosome as the template to repair the break.
Wait, so the gene drive actually copies itself onto the other chromosome, ensuring that basically all offspring inherit it.
That's the goal. Instead of 50% inheritance, you approach 100%. And the experimental results, well, they're pretty striking. In fruit flies, Drosophila, they showed a modified gene on the X chromosome spreading through generations with incredibly high efficiency, like almost perfectly.
Wow.
Which is why people are seriously discussing it for things like vector control modifying mosquito populations so they can't transmit malaria or dengue. It's potentially population-level engineering.
Yeah.
Very powerful, very controversial.
Yeah, huge implications there. Okay, shifting gears back to agriculture, plant breeding. This precise cutting ability is obviously accelerating things dramatically. We're seeing it used in major crops already, soybean, rice, tomato, wheat, maize. And you mentioned delivery, often it's agrobacterium doing the work.
Yes, Agrobacterium tumefaciens. It's this soil bacterium that naturally inserts bits of its own DNA into plants. That's how it causes crown gall disease. Scientists basically hijacked this natural ability. They removed the disease-causing genes from the bacterium's DNA plasmid and replaced them with the genes for Cas9 and the guide RNA. So the bacterium becomes like a tiny, very efficient delivery vehicle to get the editing tools into the plant cell nucleus.
Nature's little genetic engineer, repurposed.
Pretty much. And the complexity we can tackle now is amazing. Take wheat, for instance. Wheat is hexaploid. It has 3 distinct sets of chromosomes, 3 subgenomes A, B, and D.
Right, complicates things.
Massively. Often, to get a desired trait, like disease resistance, you need to modify the gene on all 3 subgenomes simultaneously. Multiplex editing.
Like the powdery mildew example in the sources.
Exactly that. Researchers found that they only achieved full resistance to powdery mildew when they managed to successfully knock out the susceptibility gene on the A, B, and D genomes. CRISPR makes that kind of simultaneous multi-target editing feasible in a way that was incredibly difficult before. It shaves years, maybe decades, off traditional breeding timelines.
And it's not just about fighting disease, right? It's modifying how the plant grows.
Absolutely. Targeting quantitative traits. In tomatoes, they've used CRISPR to target genes controlling the plant's architecture. They increased fruit size by tweaking a gene that controls the number of locules, those compartments inside the tomato.
More compartments, bigger tomato.
Basically, yeah. They also edited genes to get more branching in the flower clusters, which means more flowers, potentially more fruit, even things like making the plant bushier for better photosynthesis.
And even getting into the biochemistry, altering nutrient content.
Yes, the lycopene example is a good one. They use multiple guide RNAs at the same time, multiplexing, again, to disrupt several enzymes in the pathway that produces carotenoids.
Okay.
By knocking out a gene that converts lycopene, the red pigment, into beta-carotene, they got the plants to accumulate much more lycopene. Result, darker red tomatoes, potentially higher in antioxidants. It shows you can fine-tune metabolic pathways in really specific ways.
Which brings us right back to that regulatory question. Because initially, using agrobacterium, you do integrate foreign DNA, the Cas9 and gRNA genes, into the plant's genome. And normally that integrated foreign DNA is the trigger for classifying something as a GMO, needing extensive regulatory review.
That's the core of the regulatory puzzle. But crucially, the Cas9 and gRNA genes are usually inserted at a random spot, separate from where the actual edit happens. So the clever part is what happens next. You grow that first generation of edited plants, then you let them self-pollinate or cross them. In the next generation, because of how genes segregate during reproduction, you can find individual plants that have inherited the desired edit, the mutation you wanted, but have not inherited the Cas9 and gRNA genes. They've been segregated out, bred away.
So the final plant, the one that might go to market, has this specific DNA change you made, but absolutely no trace of the foreign Cas9 or gRNA genes that made the change. It's edited, but not technically transgenic anymore.
Exactly. Edited, but not transformed in the traditional sense. And our sources point out that, from a chemical or genetic standpoint, that final plant is often indistinguishable from one carrying a mutation that arose naturally, or one induced by older methods like chemicals or radiation.
Which creates this huge regulatory gray area. Is it a GMO or not? If it looks identical to a conventionally bred plant, how do you regulate it?
That's the debate. Many argue it should be treated like a conventional mutation, potentially avoiding the complex, costly deregulation process for traditional GMOs. But others are more cautious.
And this issue of the tool disappearing is even more pronounced in other systems, like mammals, right? The sources mentioned the controversial case of human embryo editing.
Yes. In animal cells, often the Cas9 protein and the guide RNA are introduced in a way that they only stick around for a short time. They get into the cell, make the cut, trigger the repair, and then the Cas9 and gRNA molecules themselves are degraded by the cell. They vanish.
So the editing happens,
Yeah,
and then the editing machinery just disappears without a trace.
pretty much. Unless you knew beforehand that editing was attempted or you knew exactly what tiny sequence change to look for, the edit itself might be untraceable back to the CRISPR process. It really changes the game for oversight and verification. How do you regulate something when the tool leaves no fingerprints? John Schumenti wasn't wrong comparing CRISPR's impact to PCR. These are foundational shifts. And the accessibility means it's everywhere, forcing regulators to think not just about the final product, but the process itself.
That accessibility, like we said, really sharpens the need for careful thought. We have this incredibly powerful tool capable of precise, potentially untraceable changes in plants, animals, maybe even us. So we've traced the history, that key shift to RNA guidance, the power of NHEJ for knockouts and HDR for precision edits, and these huge applications from gene drives to faster crop breeding. Which leaves us with this final thought for you. If an edited plant with the Cas9 gene gone is chemically identical to a plant with a natural mutation, how should we as a society decide what counts as genetically modified? And maybe more importantly, how do we ensure responsible innovation when the very tool used can vanish without a trace? Something to ponder, especially as these technologies get closer to your dinner table.